Hello, in this video I'm going to talk about the ways we can move substances into and out of a cell. I'm going to concentrate on active transport and bulk transport in this video. So active transport is the opposite of passive transport. With passive transport, we were moving substances with the concentration gradient. So we were moving things from an area of high concentration towards an area of low concentration while going across the cell membrane. With active transport, we're moving materials against the concentration gradient, right? And because we're going against this natural energy force of the environment, we have to supply energy. So active transport requires ATP energy on the part of the cell. It also requires proteins in the cell membrane to help transport the substances. So active transport cannot occur in a simple fashion going right across the phospholipid bilayer. We need a protein in the cell membrane to help the process of active transport along. So going back to my example that I mentioned in a previous video about kayaking in the St. Clair River, right? If you put your kayak in the St. Clair River and you flow south towards Lake St. Clair, that's akin to passive transport. But if you try to go upriver and go against the flow of the water towards Lake Huron, that's like active transport. You have to supply a lot of energy because you're going against the water gradient. There are two types of active transport. The first type is called active transport. I know that sounds stupid, but I don't get to name things in this class. The second type is called secondary active transport. What makes this easy is that we're not really going to talk about secondary active transport in our class. So we're just going to talk about active transport. So a couple examples of active transport include glucose transporters and ion pumps. So glucose transporters are what they sound like. They are proteins in the cell membrane that are transporting glucose. Now don't forget, this is active transport, so we have to have a protein in the cell membrane. That's our transporter. And we're moving the glucose from an area of low concentration to an area of high concentration. And we have to supply energy in the form of ATP to do this because we're defying the concentration gradient. Another example of an active transporter are ion pumps. So we're going to look at two examples, hydrogen ion pumps and the sodium-potassium exchange pump. So in this picture here, we can see in red the hydrogen ion pump. It is made of a protein that's embedded in the cell membrane. So the job of the hydrogen ion pump is to pump protons, aka hydrogen ions. Now don't forget, we're going to be moving these hydrogen ions against their concentration gradient. So if you notice on the side of the screen, we have a little triangle representing the gradient for the hydrogen ion. So in this case, the hydrogen ion is very highly concentrated outside of the cell. It's lower inside the cell. According to diffusion, the hydrogen ions want to enter the cell. But with active transport, we're going against diffusion. So we're going to move these hydrogen ions out of the cell where they are scarce towards the extracellular space where they're abundant. We're going against diffusion, so we have to supply ATP energy to do this. Notice the word pump here, right? A pump implies energy is involved. So we definitely have to have energy to do this. Another very common type of pump in cells in the human body are the sodium-potassium exchange pumps. Almost all the cells in your body have sodium-potassium exchange pumps, and many, many, many of them. Like any other pump, it's made out of protein, and the protein is transporting substances across the cell membrane against the concentration gradient. Sodium-potassium exchange pump implies that we're moving two things here, sodium and potassium. Exchange tells us that they're moving in opposite directions. Okay? So the sodium is not moving the same direction as the potassium. Okay? A lot of words on this slide, but let's go to a picture that kind of explains the same concept. All right, so in this picture here, first let's take a look at the side of the screen where we can see our concentration gradients. Notice that the sodium is concentrated on the extracellular side, meaning there's lots of sodium outside of cells, whereas the potassium has the opposite concentration gradient. There's very little potassium outside of our cells. Most of the potassium is found inside of our cells. Now in the cell membrane here, we can see this brown colored sodium-potassium exchange pump. So the job of the sodium-potassium exchange pump is to move sodium and potassium against their concentration gradients. So the sodium is concentrated outside, so we're going to be moving more sodium outside. The potassium is concentrated inside, so we will be moving more potassium inside. We have to supply energy to do this, and you can see our blue-colored ATP that's being used as the energy supply for this process. Another important thing to note about the sodium-potassium exchange pump is that it doesn't move sodium and potassium at a 1-to-1 ratio. It moves them at a 3-to-2 ratio. We are going to move 1, 2, 3 sodium ions outside the cell for every 1, 2 potassiums that come into the cell. Right? So it's not a 1-to-1 ratio, it's a 3-to-2 ratio. And this ratio, believe it or not, becomes really important when we start talking about how neurons work and how muscle cells work. Because this 3-to-2 ratio means we end up with 3 positives out for every 2 positives that come in, and that creates a charge difference across the cell membrane. This is a transmembrane potential, and like a battery, that transmembrane potential can be used to do work. In the last part of this video here, I want to talk about a concept called bulk transport. Technically, bulk transport is classified as a form of active transport in that it does require energy on the part of the cell, but no book's really classified as an active transport mechanism because it's so different than the other forms of active transport. It's very different from passive transport too. With bulk transport, you're removing very large amounts of substances either into or out of the cell using membrane-bound little bubble-like structures in the cell called vesicles. There are 2 main types of bulk transport based on the direction of the movement of the substance. Endocytosis is bulk transport into the cell. Exocytosis, as an exit, is bulk transport out of the cell. So let's take a look at some pictures of these concepts to understand this better. So at the top of the screen, we see 3 pictures depicting the 3 different types of endocytosis. So we have phagocytosis, pinocytosis, and receptor-mediated endocytosis. We won't study this one in this class, this last one, so you can feel free to ignore it. We're going to concentrate on phagocytosis and pinocytosis. In both cases, we're bringing substances into the cell. The substance gets wrapped around by the cell membrane, and then one side of the cell membrane meets the other side of the cell membrane, and it pinches off and it becomes a vesicle inside the cell. So the mechanisms of phagocytosis and pinocytosis are the same. These 2 are different in terms of what is actually coming into the cell. If we're bringing in larger particulate matter, we call it phagocytosis. Phago means food. This is like a cell eating. Pino means to drink. Pinocytosis is bringing in mostly liquid and maybe a few small solutes in that liquid. So pinocytosis is the cell drinking and phagocytosis is the cell eating. These substances with these vesicles will then later be processed by other organelles in the cell. Basically, the stuff will be used for food. Exocytosis is the opposite of endocytosis. It's pretty much exactly the opposite. So if you look at these pictures, they look identical, but it's the direction that's different. With exocytosis, we have a vesicle inside the cell. The vesicle moves towards the cell membrane, and the membrane of the vesicle fuses with the cell membrane, and when that occurs, the contents of the vesicle is just released outside of the cell. Exocytosis is one of the ways we release neurotransmitters by our neurons and release certain hormones. So all of these processes are important for the cell.